Hi, my name is Eleanor, and my research project is the effects of storage temperature and exogenous ethylene exposure on the ripening rate, quality, and glucose level of post-harvest ripened tomatoes. As many of you may know, tomatoes have become an increasingly popular food that is used in cooking. But what you might not know about tomatoes is that at their immature and unripened stages, they start off green, and slowly through the production of ethylene and lycopene pigment synthesis, these tomatoes start to ripen and turn red. This ripening process can be characterized visually through color change. So right here. These color changes also have a corresponding hue angle, which is a quantitative measurement of tomato ripening stage that can be made with devices like colorimeters. The typical red tomatoes that we buy from commercial grocery stores are usually vine ripened, meaning that they were harvested during stage 2 to 5, once they started showing signs of color change to red. But all around the US, and especially locally in Minnesota, once the weather gets cold, the green stage 1 tomatoes that have not yet begun to turn red and ripen are not harvested, because these post-harvest ripened or green harvested tomatoes are far less preferable to vine ripened tomatoes in both quality and flavor. With that being said, the purpose of my study was to find proper conditions to store and produce quality post-harvest ripened tomatoes in order to minimize tomato crop waste and fulfill growing demands for tomatoes. During storage, some conditions that largely affect tomato ripening, quality, like mass loss and decay, and tomato glucose levels are temperature and exogenous ethylene exposure, which trigger hormonal signals in the tomato to speed up and start its own ethylene production and ripening process. Optimal temperatures for tomato quality and ripening tend to fall around 10 to 25 degrees Celsius, while the most effective conditions for tomato ethylene production fall around 15 to 20 degrees Celsius. So now that we know about some different factors that affect tomato quality during storage, the research question I formulated was, what are the effects of temperature and exogenous ethylene exposure on the ripening rate, mass loss and decay, and glucose level of tomatoes? From previous research and data, my hypothesis was that higher temperatures and more exogenous ethylene exposure will increase the ripening rate of tomatoes while maintaining tomato quality and glucose levels. So now to my methodology. So to test my hypothesis, I set up 20 tomatoes each in 12 airtight cardboard boxes that all underwent a different combination of temperature, 4, 15, or 25, or 22 degrees Celsius, an amount of exogenous ethylene exposure, which was produced by either 0, 1, 2, or 3 bananas. As you can see in the photo, different sized tomatoes were split randomly across the 12 conditions. And for consistency, bananas and tomatoes were stored on opposite sides of the box with no contact. To measure for tomato ripening rate, the hue angle of the greenest area on tomatoes was measured daily with, once again, a chromometer. And the number of tomatoes that ripened throughout the duration of the 28-day test was recorded. This data was analyzed with linear regression, t-test, and one-way ANOVA. To measure for tomato decay and mass loss, I counted the number of decayed tomatoes in each condition and also took them out of sample groups so they wouldn't affect the other tomatoes. Percent mass loss was calculated in each group by collecting data on the tomato's daily mass change. I used t-test and one-way ANOVA for the statistical analysis. To measure for ripened tomatoes' glucose levels, individual tomatoes were blended in a 6-fold water dilution and then centrifuged in 1 milliliter vials for 5 minutes to separate the liquids from the solids. The tomato extraction samples were then tested for glucose with glucose test strips. Again for statistical analysis, I used t-test and one-way ANOVA. Now looking at the results of my data and statistical analysis. Tomatoes in 22 degrees and 15 degrees Celsius ripen significantly faster than tomatoes in 4 degrees Celsius. The daily percent mass loss of tomatoes in 15 degrees Celsius and 4 degrees Celsius were significantly lower compared to tomatoes in 22 degrees Celsius. Additionally, Only 2 tomatoes in 15 degrees Celsius were rotten, while tomatoes in 22 degrees and 4 degrees Celsius had 9 and 8 rotten tomatoes respectively. While I was able to reach a conclusion on the effects of temperature on tomatoes, I was unable to draw any conclusions about the effect of exogenous banana-derived ethylene exposure on tomatoes, because I was unable to control and regulate a constant amount of ethylene released by the same amount of bananas throughout the groups. Bananas released unconstant amounts of ethylene in different temperatures because banana ethylene production is temperature dependent. However, in my results, I did make note of another trend I saw between tomato size, and its glucose levels. Overall, there was a strong trend for larger tomatoes having higher glucose concentrations than smaller tomatoes. Looking at the larger picture, how did temperature and exogenous ethylene affect post-harvest ripened tomato ripening rate, mass loss, decay, and glucose level? First, we can eliminate exogenous ethylene, because while it may be able to affect tomato qualities during storage, the results were not effective in this study due to the limitations of bananas as an ethylene source. Next, we can draw the conclusion from the results that temperature does indeed affect tomato ripening rate because of its effects on the hormones and enzymes required in lycopene and ethylene synthesis. Higher temperatures also cause easier bacterial growth, while lower temperatures weaken tomato wall cell structure. Both of these increase the decay in tomatoes. Thus, my hypothesis was only partially supported. Finally, while both the properties of tomato glucose level and mass loss have been explored in this study, no conclusion or reasons for how temperature or exogenous ethylene significantly affected them could be made. Thus, this gray question mark box represents other factors besides temperature that could have caused tomato mass loss and why glucose levels were affected by tomato size. To find these unknown factors, future applications could focus solely on examining how different tomato sizes affect tomato glucose level, and future applications could also adopt a different approach to studying how temperature and other factors affect tomato mass loss. Thank you.